Today, I'll be doing a mind map on cells and the structures within them. All cells can be grouped into two categories, prokaryotes and eukaryotes. Eukaryotes make up multicellular organisms such as plants and animals, where its structure varies depending on the organism. What you see here is an example of a eukaryotic cell for animals. On the other hand, prokaryotes are unicellular organisms, with one example being bacteria. I will show you the structure of a prokaryote later on, and we can compare the differences between the two. However, regardless of the type of cell, cells share four common features which classify them as living cells. Firstly, every living cell is surrounded by its own membrane, which separates its contents from the outside. Secondly, all cells contain genetic information which stores instructions for the cell's activities. These activities mainly come in the form of enzyme-catalyzed reactions which take place within the cell. Lastly, each cell has its own energy release system which powers all cellular activities. Eukaryotes contain organelles, which are specialized structures within living cells that are surrounded by its own membrane. There are three organelles that have double membranes: the nucleus, mitochondria, and chloroplast. A basic structure of chloroplast looks like this. We'll look more into chloroplast in my mind map on photosynthesis. The first organelle we have is the nucleus, which is essentially the brain of the cell. It stores most of the genetic information within the cell in the form of DNA, where this information controls cellular activity. A nucleus is found only in eukaryotic cells, where prokaryotes do not have a nucleus to store their genetic information. Next, we have the rough endoplasmic reticulum. The RER is a network of flattened membrane sacs called cisternae, which is continuous with the nuclear envelope. It is identified by black dots all over the membrane, which are ribosomes. This feature is helpful when identifying the RER under a microscope. The function of the RER is the formation of secondary and tertiary protein structures from the amino acid chain synthesized at the ribosomes. As discussed in my mind map on DNA, a polypeptide chain, also referred to as an amino acid chain, is synthesized at the ribosomes through translation. Once formed, the protein chain, currently with a primary structure, moves into the ER, where secondary and tertiary structures begin to form. This is the first thing that happens. In addition to this, the protein is also tagged with a biochemical marker which indicates its next destination, the Golgi apparatus. Once more complex structures have formed, the protein is packaged in vesicles pinched off from the RER membrane and begins travelling to the Golgi apparatus. The Golgi apparatus is characterized by its disc shape, and like the RER, it consists of cisternae. The main function of the Golgi apparatus is to chemically modify substances made by the ER, process them, and package them in vesicles for secretion out of the cell. When vesicles from the RER reach the Golgi apparatus, it enters via the cis phase of the Golgi apparatus and releases its contents. These substances are modified further, which can include formation of more complex structures and synthesis of macromolecules. For example, proteins that come from the RER can form more complex tertiary or even quaternary structures with multiple proteins. Macromolecules such as glycoproteins and lysosomes can also be synthesized. Once completed, the products are packaged in vesicles pinched off from the trans phase, which is transported, usually, for secretion out of the cell. However, there are some products produced for use within the cell, as is the case for lysosomes. Lysosomes are organelles within the cell that is formed by fusion of vesicles from the trans Golgi. They contain enzymes whose function is for digestion within the cell. Lysosomes digest three main things: food vacuoles, walnut organelles, and bacteria. The last big structure in the cell is the smooth endoplasmic reticulum. It is made up of the same flattened membrane sacs as that of the RER. The difference is that the SER is not continuous with the nuclear envelope, and it does not have ribosomes on its membrane. The SER's main function is the synthesis of lipids and steroids, as well as the conversion of harmful substances to harmless ones, through processing toxins, for example. The only organelle in animal cells, apart from the nucleus, that has double membrane is the mitochondria. The reason for its double membrane is the formation of cristae, which is essential for the electron transport chain and chemiosmosis in respiration. This will be covered more in my mind map on respiration. Respiration is what produces energy in the form of ATP, and the mitochondria is essentially the powerhouse of the cell, as it contains this energy release system. Another organelle that you will come across are the centrioles, whose main role is in cell division. You can check out my mind map on cell division here. Lastly, we have talked about ribosomes, whose function is the synthesis of proteins. They are located either on the RER or lying freely in the cytoplasm of the cell. However, do note that ribosomes are not organelles. As you can see, eukaryotic cells have a pretty complex internal structure. This is very different to prokaryotic cells, which we will see. The largest differentiating factor of eukaryotes is its compartmentalisation, as seen by the numerous compartments referred to as organelles within the cell. The purpose of compartmentalisation is for boosting efficiency of cellular activities, which is beneficial for more complex eukaryotes but unnecessary for the simpler prokaryote. The advantages of compartmentalisation include: Number 1. Enzymes and substances for different processes are concentrated in smaller compartments, which allow the reaction to occur faster. Number 2. Substances harmful to the cell are kept within their compartments, as is the case of digestive enzymes being kept within the lysosome. Number 3. Optimum conditions can be maintained within each compartment based on the processes taking place. And Number 4. Organelles and their contents can more easily move around within the cell. Now to prokaryotes. Compared to eukaryotes, prokaryotes are a lot less complex. Some features that immediately differentiate them from eukaryotes is the presence of pili. Pili are protein filaments protruding from the cell wall. The purpose of pili is for cell-to-cell adhesion and conjugation, which is a process when two cells exchange DNA with each other. Another thing you will notice is the flagellum, whose base is embedded in the cell wall. The purpose of it is for cell movement, where the flagellum can rotate to propel the cell. Flagella are not only present in prokaryotes but some eukaryotes as well. Contractile vacuoles are organelles often found in unicellular organisms. They are specialized vacuoles that maintain the osmotic balance in the cell. It does this through osmoregulation, where it fills up with water and expels it from the cell. The nucleus is the region in the prokaryote containing genetic material. Prokaryotes usually only have one molecule of naked DNA, and unlike eukaryotes, it is not stored in the nucleus. The last thing we have to talk about are stem cells. It is important to know what they are, as they can come out as a nature of science inquiry. All cells begin as undifferentiated cells, which then specialize for a specific function. Stem cells are undifferentiated cells that still have the capacity to divide and to differentiate. The advantage of stem cells is that they are able to endlessly divide and differentiate. This is useful for the repair and regeneration of tissues, for example. The main idea is that with stem cells, you are able to differentiate to a specific cell of your choice. This is the main focus of stem cell research. The ethical consideration of this form of research, however, is in the destruction of human embryos to harness embryonic stem cells. Compared to adult stem cells, embryonic stem cells can differentiate to form more types of cells, making them more useful and more desired. Furthermore, it is more difficult to locate and extract stem cells from adults than it is for embryos. This leads to ethical dilemmas, which is something you should consider. This brings me to the end of my mind map on cells and the structures within them. I hope you find it useful and have a good day.